Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about the transformation, including the principle, the protocol, and the troubleshoot. Let's begin. So first, you have to know that different kind of the E.coli strains that used for the transformation. So those are BL21, that is a protease deficient genetically engineered, and the DH5 alpha, that is genetically engineered with REC-A1 mutation. Along with that, also other kind of E. coli strains are used for the transformation as well. But these two strains are mostly common that used for the transformation. The BL21 that is used for the protein expression, and the DH5 alpha that is used for primarily for the plasmid transformation. So next have a look at what is transformation. So transformation is the process of incorporation of the recombinant plasmid into the bacterial cell, and that plasmid contain our target gene. So that plasmid is known as the recombinant plasmid. So next have a look at enter protocol to conduct the process of the transformation. So first you have to take out the competent cell of the E. coli, and then you have to put on ice to thaw the cell for 5 minutes. And then you have to add the plasmid or the PCR product onto the competent cell and then mix properly and leave it 30 minutes on ice. And the plasmid and the competent cell ratio, it should be 1:10. It means that if you have the 100 microliter E. coli, then you can use around 10 microliter plasmid that containing 5 to 10 nanogram of the plasmid, or you can use little more, that's no problem. And next process, the tube containing your plasmid and the competent cell, that you have to put on 42 degrees centigrade for 3 to 4 minutes water bath. And immediately you have to, after 4 minutes, you have to put the tube on ice for 90 seconds. This is just for sudden temperature drop that makes pore into the cell membrane. And next step, you have to take out that tube, and then you have to put onto the rack, and then add 5 microliter sterile LB media. But remember that in this step, no need to add any antibiotic in that LB media. And then mix well, and then put incubator for the incubation, 37 degrees centigrade, 150 rpm for 45 minutes. And this step is for to grow the antibiotic resistance gene in the bacteria. And next step is to that you have to plate that bacteria onto the agar plate, and agar plate must contain antibiotic. And basically the ampicillin and carbonicillin, that concentration, working concentration would be the 100 microgram per ml, and the stock can prepare for ampicillin and carbonicillin around 100 mg per ml. So if the plasmid, if you use the plasmid for the transformation, then you can use only for 200 to 300 microliter of this bacteria. But if you use like PCR product that containing mutation, then you can add all the bacteria. So after adding that bacteria onto the agar plate, you can incubate it for 37 degrees centigrade for 16 to 17 hours. And after 16 to 17 hours, you might get these clones. So if you use plasmid for the transformation, then you might get a lot of clones. But if you use PCR product that containing mutation, you might get very less number of the clones. Even I got sometimes single clones. That not be worried about it. So in this step, you can check these clones by colony PCR, whether that plasmid is that clones is the right clones or not. So next is the picking clones. So after getting the clones, you have to use the sterile white yellow tips to pick the clones. And for the mutation, recommending that around 10 clones you can pick at once at the same time in different tubes. And the tube, it should be the round bottom tube, that is like it's suitable for the bacterial growth. So after picking the clones, you have to use around 3 to 5 ml LV with antibiotic, around 100 microgram per ml in this LV, and then you have to add these clones into this LV. But remember that this LV must contain the antibiotic. And next is the incubate that bacteria for 37 degrees centigrade, 150 rpm for 16 to 17 hours. And next process is sequencing to check that your desired mutation is there or not. If it is like plasmid or just plasmid, then you don't need to check the sequence. But if you have the mutation, then you must check by sequencing. You must confirm by sequencing. So you can check the clone is right size or not by this step. You can do this step, the colony PCR, or you can do this step. So you can also use your bacteria to amplify the DNA. Then you might get it if you run the DNA gel, so you can understand that which is the right clones or not, that clones is whether it's right or not. So for example, if you have like 4 clones, and then if you after amplification, if you run the DNA gel, then you understand that this clone is the right size and this clone also right size, but there are, you can see the number 2 and number 4, there is no amplification. So it means that number 2 and number 4, that is not the right clones. So that don't contain the target gene, target plasmid, right? So you need to use this number 1 or number 3, this both for sequencing. So troubleshoot. Also there are some troubleshoot you might get. So first of all, that I'd like to mention the satellite clones. That is like if you incubate your agar plate for longer time, then you might get this kind of small kind of clones. You can see this kind of small kind of clones, and this is for the large clones that secrete beta lactamase enzyme to degrade the ampicillin and leading to reduction of the ampicillin in that particular area. As a result, wild type E. coli begin to appear and grow, and this kind of wild type clones, that is known as the satellite clones. So remember that small clones or satellite clone contains no target gene at all. So to avoid this kind of satellite clones, you have to check that antibiotic stock is too old or not, or the antibiotic concentration, if it is very low, then also you might get this kind of satellite clones. And also when you add this antibiotic onto the agar plate during preparation of this agar plate, then you must not add this antibiotic in hot condition. And also you have to stir it, mix it properly, the antibiotic, and also don't grow the transformated plate very longer time period. And next is that sometimes you might not get any clones onto the plate, and there are some reason that you can check the competence cell, maybe it is very old, so you can use the new one, or the correct antibiotic you used or not, you have to check it. And next is if you have like too many small clones grow on the plate, also you might like antibiotic stock, you can check, you can use the fresh antibiotic stock, or maybe the antibiotic concentration is very low, or you can stir the antibiotic in the growth medium properly. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and please don't forget to subscribe my channel. Thanks for watching.